Okay, let's get this show on the road here. Hello, everyone, and welcome to The Current. This is the North Central Region Water Network's Speed Networking webinar series. As we have been saying for the past few months, thank you. We are so grateful that you are all out there taking care of our land and water, even as you are taking care of yourselves and your loved ones under so many different circumstances. We hope you are all staying safe and healthy, and hope we are doing our own small part to make your jobs a little bit easier. I'm Rebecca Power, joining you from the University of Wisconsin-Madison, and I will be your moderator. Since some of you may be new to the North Central Region Water Network, we are an extension-led collaboration among land-grant universities and our partners in 12 upper Midwestern states. As it says on this first slide here, our topic this month, our timely topic, is harmful algal blooms. And with summer in more than full swing, we're sweating here. I'm guessing most of you are where you are. Many folks are hopefully enjoying spending time out on the water. And with harmful algal blooms, those can put a damper and some safety issues on with our summer fun. So today we'll hear about two network-funded projects, one led out of the University of Missouri that is bringing teachers and students into community or citizen science around harmful algal blooms. Some interesting results there from an outreach perspective, and some interesting HABs research results as well. Our other project is led out of Iowa State University and provides an update on a collaborative project between the North Central Region Water Network and the United States Geological Survey, or USGS-funded water resource research institutes in the North Central region. So that project is really trying to expand translation of HABs research into products that can improve HABs outreach and management. Before we introduce our speakers, a few notes to you all about how these sessions work. Please submit your questions for presenters in the chat box. That chat box is accessible through the little purple tab that is in the lower right-hand corner of your webinar screen. We'll have a dedicated Q&A session after the last presentation. These presentations are going to be about 15 minutes each, maybe a little bit more. So we're hoping to have plenty of time for questions. If you're having audio issues, a phone-in through your computer audio, a phone-in option can be accessed by opening up what Blackboard Collaborate calls the session menu in the upper left area of your screen, and you can select the option use your phone for audio. We do record these sessions. They will be available for later viewing at northcentralwater.org and learn.extension.org. You can also follow this session on Twitter. We've got some live tweeting going on, and please, we would love it if you would share this information with your colleagues that you think might be interested. All right, so our presenters for today, our first presenter, Emily Kinzinger at the University of Missouri. She is a graduate research assistant with the University of Missouri Limnology Lab. And we are really pleased to be able to feature a student on this webinar. As you know, these students, including Emily, are our next generation of water resource professionals, and obviously they, like all of us, have been going through a lot of challenges with changes in campus environments and lab, convening issues and all that kind of stuff. So we're happy to be able to feature Emily's work today. And second, Melissa Miller, the Associate Director of the Iowa Water Center at Iowa State University. So you'll hear a little bit more, see a little bit more about the background of those folks in just a minute. And that minute has arrived. So here's Emily's bio. As most of you know, I don't read through these verbatim and let you take a look on the screen. She is an MS student working with Dr. Rebecca North at the University of Missouri Limnology Lab. This project is a partnership between our research colleagues at the University of Missouri and participating states, as well as our extension colleagues. And Dan Downing from the University of Missouri is our network leadership team member that's been supporting, one of the people supporting this project at the University of Missouri. So a little bit more there about Emily and the background that she comes to us with. And with that, we'll turn it over to Emily. So thank you so much, Emily. Go ahead.
Thank you for that introduction. Yeah, so I'm Emily Kinzinger, and just to reiterate, I'm a master's student at the University of Missouri Limnology Lab. Today I'm going to be talking to you about the Reservoir Observer Student Scientists program for year-round harmful algal bloom monitoring. So we'll go over some background on HABs and what are harmful algal blooms, and then I'll talk to you about the Reservoir Observer Student Scientist, or ROS program. We'll go over some research questions that the ROS program is helping us to answer, and we'll go through some data that we have analyzed from the samples that the students have collected. And just a sneak peek, we have found that chlorophyll A, which we're using as a proxy for algal biomass, does not vary by season. And from the algal toxins that we're looking at, microcystin concentrations are higher in the summer months, but cylindrospermopsin concentrations do not differ from season to season. And we are not seeing any relationship between algal toxins and chlorophyll A, nor is there a relationship between the toxins themselves. So for some background on HABs, eutrophication, or the over-enrichment with plant nutrients to our surface waters, is a water quality issue on a global scale. And this is largely due to anthropogenic inputs of nutrients from things like ag and urbanization runoff. Increased nutrients in our surface waters, specifically nitrogen and phosphorus, can result in excessive algal growth, increased turbidity in our lakes. They can deplete our subsurface oxygen, leading to fish kills, reduced recreation, declines in property values, and all of this can lead to economic losses in excess of $2.2 billion annually. And finally, increased nutrients can also result in a shift in species composition in our lakes from more favorable algae types to cyanobacteria, which is going to be the focus of this presentation. So cyanobacteria can form these dense green mucilaginous scums on our lakes and reservoirs to where it almost looks like someone dumped in a can of green or blue paint. They have taste and odor problems, which can be especially problematic for reservoirs that are used for drinking water because these compounds are hard to get out of the water. They can shade subsurface vegetation, and they can also potentially be toxic. So about 75% of blooms are dominated by species that have the ability to produce toxins. There are a few different kinds of algal toxins, but the two that we're looking at in this study are microcystin and cylindrospermopsin. These are both liver toxins, and microcystin can also act as a skin irritant. Exposure is harmful to both humans and animals, and we can be exposed through inhalation or ingestion during recreation, and a lot of animals are exposed from drinking contaminated water. It can lead to both acute and chronic impacts. I'm sure a lot of you remember last summer whenever there were many dogs being poisoned by drinking lake water contaminated with algal toxins, and this affects a lot of livestock as well. There are many, many theories as to why cyanotoxins occur in our lake water. A few of them relate to positive relationships with light, higher water temperature, in different relationships with nutrients, but that is pretty complicated and there are lots of different theories and no consensus at this time. And so blooms like it hot. They really thrive in the summer months whenever there's lots of sunlight, warm water temperatures, and stable lake conditions. And a lot of recreation also happens in the summer months, so people are out and about on lakes, they're noticing what's going on, and that's when most of our monitoring takes place. However, our shoulder seasons, so spring, fall, and even winter, are understudied, but the available research does indicate that blooms in these seasons can produce toxins. So it's really important that we understand what's happening year-round to have a good picture of how to manage our lakes and reservoirs. And so to help us monitor lakes year-round, we created the ROS program. So this is a novel water quality monitoring program where we work with high school students to not only teach them about HABs, but to also collect year-round water samples. The way this works is representatives from our lab, so that has been myself in the past and a lab technician, go to different schools and we teach the students about limnology and their local reservoir, and then we actually go out to the lake with them and teach them how to sample for water. We take the water samples back to their classroom and we filter it and process it for preservation, and then the samples are analyzed back at our lab at NU. And we provide all of the equipment needed for the students to do this. So far, we have implemented the ROS program at 4 different high schools, and so students have been sampling Bethel Lake here in Columbia, Missouri, Cheney Reservoir in Maize, Kansas, Lake Sakata in Waterville, Minnesota, and Black Hawk Lake in Lakeview, Iowa. We started this as a pilot program in the fall of 2017, and since then we have trained 300 students. And so that's 300 students that have not only learned about water quality and HABs, but they've also gotten to take part in a real scientific study and learn things about recording good observation, or properly fill in data sheets and stuff like that. One of the highlights of the ROS program for me is the group of students that have been sampling Cheney Reservoir in Maize, Kansas. So this group of students has taken the initiative to do some of their own research on the reservoir, and they actually presented their work on some microstratification in the reservoir last fall at the Great Plains Limnology Conference, where they won the Best Undergraduate Presentation Award. And then my favorite thing is that they took the knowledge that they learned about HABs and they travel to other high schools throughout Kansas and teach different FFA groups about harmful algal blooms. So we've got some peer-to-peer teaching of the next generation of farmers about how to properly manage nutrients and help prevent HABs in the future. And so some of the research questions that we're hoping to answer from samples that these students collect are hot moments in cyanotoxin production happening in the cold. And we want to know how the presence and concentration of cyanotoxins relate to different physical and environmental drivers. And lastly, we want to know if cyanotoxins are correlated with chlorophyll A, which, like I said, we're using as a proxy for algal biomass because algal counts are time-consuming and very expensive. And so the samples the students collect are analyzed for total nitrogen, total phosphorus, chlorophyll A, the algal toxins, microcystin and cylindrospermopsin, and total suspended solids. And I just want to note that the figures I'm going to show you, the data is just for Bethel Lake and Cheney Reservoir. I was on maternity leave for a while, and then our lab shut down due to COVID-19, so we have a bit of delay in analyzing the rest of the samples from last school year. And our samples are divided based on summer and non-summer seasons. So summer has been defined as the period of stable stratification for Bethel Lake, and non-summer then is the rest of the year. And it's worth mentioning that neither of these lakes sustains ice cover throughout the winter. So this first figure shows season on the x-axis and concentrations of chlorophyll A on the y-axis. And what we see here is that chlorophyll A concentrations do not differ from season to season. And so this is counterintuitive. Since blooms like it hot, we would have expected to see large summer concentrations, but we're not seeing a significant difference between the two seasons, thus highlighting the importance of year-round monitoring again. And here we're taking a look at how the concentrations of toxins vary from season to season. So this top figure shows concentrations of microcystin on the y-axis by season. And here we are seeing a significant difference in microcystin concentration. So they are higher in those summer months. Whereas cylindrospermopsin on the bottom figure, those concentrations do not vary significantly from season to season. In these figures, we're looking at the relationship between chlorophyll A and our toxins. So we did not actually detect any kind of relationship from either microcystin on the top figure or cylindrospermopsin on the bottom figure and chlorophyll A. And what this tells us is that our strategy of just telling people to stay out of green lakes might not be enough to prevent them from being exposed to algal toxins. And these concentrations are relatively low, but it's definitely important to note that algal toxins can be present in lakes whenever they aren't very green. And something else that's interesting on this bottom figure that shows cylindrospermopsin. So the black triangles represent the non-summer samples, and you can see that our highest concentrations were actually found in the non-summer months. and not summer like you would think. So here we're looking at the relationship between the two toxins. We wanted to see if there was anything there, but once again, there's no relationship between cylindrospermopsin and microcystin. And our last figure, so this is just for Bethel Lake, and we're looking at how the toxin concentrations vary from year to year. So on the x-axis we have month, and then on the left y-axis we have microcystin concentrations, and on the right y-axis we have cylindrospermopsin concentrations. So you can see those blue circles are cylindrospermopsin, and so our monthly average samples had higher concentrations in late fall and early winter of 2017 to 2018, but then kind of leveled off to below detection limits. And so this highlights how toxin concentrations can vary from year to year and shows the importance of long-term year-round water monitoring. So some conclusions from all that. Chlorophyll A is not always higher in the summer months like we would anticipate, and microcystin concentrations were significantly higher in the summer, but we did not see any significant seasonal differences in concentrations of cylindrospermopsin. And we did not identify a relationship between toxins and chlorophyll A, so we need to be careful to not just tell people to stay out of green lakes to keep them safe. And toxins can be present in the winter months, so it's important that we keep monitoring lakes year-round. And finally, the ROS program has been invaluable in helping us collect year-round water quality data, and we are very thankful for the students that have participated. We are continuing the ROS project. I know things might look a little different this year because of COVID, but we still want to recruit any interested high schools. So if you know any teachers that might be interested in participating in this project and their school is located near a lake or reservoir, we would love if you would send along their contact information to us, and you could email that to my advisor, Dr. Rebecca North, at that email address. Thank you.
Excellent, great. Thank you, Emily, so much. And any questions for Emily, please go ahead and put them in the chat box. And again, I think this was clear, but just a reminder that those schools can be at least anywhere in the North Central region, in the upper Midwest and Northern Great Plains. And I'll just ask Emily quickly, would you entertain inquiries from states that are beyond the North Central region, or are you mostly sticking in the North Central region?
I'm actually passing off this project, but I think that they would entertain offers for other states as well.
Great, thank you so much. And Rebecca North can clarify that if that's not the case. Super. Okay, thank you, Emily. And have some good questions pouring in there, which we will hold until after Melissa Miller's presentation. And again, Melissa is the Associate Director of the Iowa Water Center at Iowa State University. She's going to be talking to us about partnering to develop research, outreach, and education resources to mitigate harmful algal blooms in the North Central region. And this is a great opportunity, I think, for folks participating to give some feedback to Melissa and the team working with her, the research and the outreach team, on some of the products that they are planning to create. So thank you so much, Melissa, and go right ahead.
Yeah, thank you so much, Rebecca. And you're absolutely right. I have an entire slide on how I'd like people to participate in the project. So good afternoon, everyone. I also want to thank Emily for talking about that project. So it's really exciting work. I was very interested in your takeaways. That's exactly the kind of information that this project seeks to incorporate into outreach materials. So last fall, my team members and I presented on the first phase of this project, which was the white paper. So some of you may have tuned in for that, or you may have already read through the white paper or seen some of the publications that we've done around that. So what I'm here to talk about today is phase 2 of the project, which is the implementation phase. So now we're working to further develop the recommendations that we made in the white paper. So I'll cover a bit of the takeaways from the white paper just to set the context, and then I'll give you some more detail about what we're working on right now, and then again, how you all can get involved. So I see that a few of our team members are on the list today, so you're all welcome to heckle me in the Q&A if I get anything wrong when talking about the project. So this group has been working together since early 2018. We had a grant from the North Central Region Water Network, and we originally set out to build this team that had representatives from each state in the region, from both Extension and the Water Resources Research Institutes. Our goal was to review the research and the outreach materials on HABs produced by our respective organizations, and then we wanted to identify the gaps and make recommendations to fill those gaps, all with the goal to move toward a more synchronized, comprehensive regional approach to mitigating harmful algal blooms. So in October of 2019, finally, we released our white paper with messaging recommendations, future research needs, and a list of potential outreach products to be developed. So I'll give you a quick overview of those key points here with the next few slides. So after reviewing the research and the available outreach materials, we decided that we needed to compartmentalize specific topics related to harmful algal blooms as it might pertain to our different stakeholders. So these key themes that are listed here are fairly reflective of the research topics from the WRRI funded research, and we think they capture a broad spectrum of HABs-related concerns. And a lot of this sounds familiar when we hear Emily talking about harmful algal blooms. So you can see on the slide, we have general HABs knowledge, identifying, monitoring, and treating HABs, human health and HABs, animal health and HABs, and then landscape nutrient management practices in HABs. So from there, we identified key messaging based on the research that's been conducted, both from our organizations, from the institutes and extension and our universities, but also more broadly from the agencies and the institutions conducting HABs research in the region and around the country. There are tons of people that are working on that because it's such an important issue, and I just want to make it clear that we are definitely monitoring the work that everyone is doing and incorporating that into our work. So all of our key messaging is separated not only by those topics that are listed, but we also did sort of a level system. So think of it like a 101, 201, 301. We have the general public. That's anybody who might encounter HABs, your average producer, a casual beachgoer. We have the engaged citizen. So here we're defining this as a person who's likely to take action of a result of the information they learned beyond themselves or their immediate family. So maybe they'll do some community organizing, or maybe they'll talk to a group of people, or they're already leading an organization that works within the community. And then we have the water professional. So that's somebody who is charged with working with HABs as part of their job. So the messaging is all contained in the white paper, and I'm not going to cover that today, but just know that we're using that messaging that we developed as a basis for much of the work that we're doing here in phase 2. We also compiled a list of future research needs based on the research that's currently being conducted, as well as needs that have been articulated by our colleagues in the research sphere. This list is intended to be a guide for future collaboration efforts among the institutes, but also for our partner organizations. These are really applied research areas. So it's information where if we had the answers to these questions, we would want to incorporate those into our future outreach information. So I'm not expecting you to read that very, very tiny print, but it's a direct screen grab from the white paper. So we made a list of products to develop, and that's where we began our work here with phase 2. So in phase 1, we looked at what we had for existing HABs outreach, and then we talked about the types of products that would be most useful to our primary stakeholder. So our primary stakeholder right here is extension and university educators. So we are specifically talking about the people who are going to go out and work with the communities that we have identified as our messaging. We want to, it's sort of a train the trainer type of idea. We want to give them the resources and materials that they'll use in their work. So there's a variety of products here. There's 9 total listed, and we knew that we wouldn't be able to tackle all of these in phase 2. So we did the implementation process by narrowing this list down and then choosing the highest impact, most feasible products to develop. Now, before I jump into these deliverables, I do want to mention that we had a bit of a delay in kicking off phase 2. As you all experienced, we had a pretty busy winter, and then right about the time that we were going to kick things off, the pandemic happened, and we went right into pandemic response. So we didn't really start this phase until May of this year. So we reorganized, we collected our bearings, we went forth with the second round. So full disclosure, this is still really the beginning phases, but that's a perfect time to be talking to you right now because this is really where we want to invite you to come in as part of this process. And we do anticipate making quick progress on these over the next 6 months or so. So as you can see here, our list went down from 9 to 5. And actually, one of these wasn't even on the original list, but that's all right. So I'm going to go over these in more details over the next few slides here. The first category of product is really a collection of products. So this is all predicated by the messaging that we developed in phase 1. We want to produce a bank of a variety of text types that are useful in the day-to-day work of our extension and university educators. So we plan to develop fact sheets for each of the topic areas. We're going to have both a finished stylized product that you can just print and use it and give it out to people, but we'll also have it available plain text in the event that any of our institutions wish to incorporate it into their own product branding scheme. So we want it to be as useful as possible. We're also developing mid-length narrative text. So we're talking like 400 to 500 words here. These are blocks of information that can be either dropped in as is, or they can be adapted for things like blog posts and newsletters, press releases. The idea behind these is that somebody could take this information that contains, evidence-based information about harmful algal blooms. They can customize it with the topical information that they need for their state or their particular situation, like a bloom occurrence. They have this product that's ready to go. They don't have to recreate the wheel and go out and search for that text. We have something that's available right there. So the messaging that we have in the white paper is actually perfect for social media. They're all bite-sized snippets of standalone information, typically, one sentence. So we're going to go through them all and edit them to ensure that they're social media friendly. And then we're going to accompany them with a hashtag that can link all of that information together. And that way all of us in the North Central region that are using this information can have a common link. So finally, we talked about having short case studies of HAB occurrences ready for the educators to pull from. So and we want to collect this by state and by topic. So this group of snapshots will be housed with these text resources. So if I think that HABs are important and I want to share information about HABs with people who are around me, I can go to this bank of case studies and I can see where this happened in my state that maybe connect me more. It's going to be more relatable for the people that I'm working with. So this is definitely an area where we would love to see your help. So when we say snapshots, we're talking, 250 words or less. We just want to know of a time that a HAB occurred in your state and what that looked like and how you responded, things like that. So the video shorts will be a collection of lightly produced videos taken in the field by the researcher that covers different topics related to HABs. So I say lightly produced here because we want to get these videos up and running quickly. We want to do it for not very much money, particularly because this is partially in response to where we're at with online education. One of the main audiences for these videos is going to be faculty that are teaching lab courses online. They need content that shows the type of analytical concepts taught in water resources. So our methodology for this will be to comb through what already exists because there have been some series that have already done some of this in the field type work. Also, we want to reach out to you all, to our networks, and we want to find folks who are willing to record themselves or somebody in their lab or in the field and send us the raw video so that we can edit it just for this purpose. The other thing I would say is if you are a water faculty or if you're teaching some sort of a water resource class and you can think of core concepts that would be useful to have a video on, let us know those as well and we can try and fill that need for you. We will have a survey that goes out to the networks that we have of water faculty, but my contact information is on the last slide and I'd be happy to add you to that list. So one of the best outcomes from phase 1 wasn't necessarily the white paper or the recommendations. Obviously, that's the meat and potatoes, but the energy to come together around one particular issue. So we felt that holding a research symposium with specific productive outcomes would be a good use of or outlet for this momentum. So we are talking about holding a virtual research symposium this December. And we chose December because we knew that there was going to be some uncertainty with how the fall would go for our university participants. Also, this is past field season. So we were hoping that December was the right time to have these conversations. So we have a few objectives for this. First, we want to introduce, play a little, this is a speed networking event right here, right? We want to do a little bit of that too. So we want our institute researchers, and we say institute adjacent because the institutes, none of them are working alone in their states. They're working in a large partnership. They're working with different agencies and universities and organizations to do the work that they're doing. So our networks, we want to introduce them to their peers from different states working on similar topics, and we're particularly talking about HABs this time. We want to continue to identify the gaps in the research in HABs. We talked about how we could develop a review paper, we could have a series of papers, we could even have a special edition of a journal. We talked about developing teams that are working on major multi-state outreach efforts that incorporate extension and outreach as part of the broader impacts. So how do we build up these teams that are working collaboratively and going after larger federal and foundation funds? And then we talked about it would be really nice for the symposium if we could identify some of those potential funding resources before, or maybe there's a unique funding source that we can about, and we have some of the abilities to do that through the Water Resources Research Act funds within USGS. So that's something that we're working on. The actual method for holding the research symposium, we've talked about doing it over the course of a couple of days. We're still pretty early in the planning for this. And each of the sessions would be, they have the ability to be standalone based on topic. So you can, dial in for all 6 sessions, you can dial in for 1 session, but there will be half of it reserved for lightning talks on current research, kind of like what Emily just did for us. And then the second half will be really focused, facilitated discussion on how we can incorporate what we've just learned into what we already know and forward for new discovery and inquiry. So then we talked about having a webinar series. And the webinar series, again, what's great about this initiative is that it's building up this partnership between the institutes in the region as well as the extension. And this is a partnership that we think we should have had for a long time, and that we can build upon in the future for lots of different topics, starting with HABs. So we talked about how we can have a webinar series that's highlighting the research that's being done, again, the research that's being funded by the institutes, as well as the institute adjacent work that's being done. So this is a way to continue to build that community of practice in the region, focusing on particular water research needs. So then finally, we have a resource web page, and this is essentially just a page that's going to host all of these resources that we're developing. It'll have all of the ready-to-use text, it'll have the videos, and it'll have links to other resources, and I'll talk a little bit about that in just a second here. So opportunities to participate, I promised you a slide. First of all, we'd be very happy to have technical reviewers for the written word text areas, especially some of those longer form ones. So if you attended the original webinar on this project, or you've heard me talk about it in any form, you'll know that our team members are not necessarily HAB scientists. We're all good communicators, but we would be glad to have folks that are working with this on a regular basis serve as a technical reviewer for the things that we put together. We're looking, again, for those real-life snapshot stories of HABs impacts in your state. We would love to have a large portion of you recording or facilitating recording of the video shorts. you can serve on that research symposium planning committee. And we definitely invite you all to attend that symposium. There'll be more information. We'll be sure to publicize that through the North Central Region Water Network channels. But right now, if you're interested in serving on that research symposium planning committee and helping us make some decisions about how we'll do it and who will be involved in presenting and the types of conversations we'll be having, you're more than welcome to join that. That's certainly not limited to our project team. You can suggest themes and topics for future webinars. We're starting with HABs because this is such a pervasive issue in the North Central region, and it's certainly a topic that we need to be discussing. However, we know that there will be things in the future that we'll want to focus on, and this will also help inform the research that's conducted at the institutes. So finally, we have contributing other resources for link on resource web pages. So something that came up in the first phase is just the understanding that there are so many agencies that have provided some really good information online regarding harmful algal blooms, regarding reporting harmful algal blooms, identifying harmful algal blooms, things like that. If you have good resources, we want you to direct people to your resources, particularly if you're updating those on a very regular basis. We understand that we have this energy to create this product right now, but in terms of being able to maintain it, that's not necessarily the job that we have 100% of the time. So if you have a resource that we would like to create evergreen resources that need to be updated, maybe not as often, but then refer people to the resources that you have. So finally, I know Rebecca put a link to the white paper in the chat. You can certainly find that at the North Central Region Water Network. Or you can contact me to participate, and I'm happy to take any questions now in the Q&A format.
Excellent. Thank you so much, Melissa. Great presentation, and thanks, Emily, again. And now we will go back for questions. And thanks to Rebecca North also for helping to answer some of those questions in the chat box. So first, we will start with Jim. For Emily, what is the cost for equipment that is provided to the schools?
I would say it can range from about $500 to $750, depending on what all you're sampling for and what you have already.
Just quickly give us an example of the things that are included in that cost.
Yeah, so that includes supplies for collecting the water samples and for filtering and processing them. So like a filter funnel assembly, Erlenmeyer flasks, lots of different plastic bottles and glass vials for them to put the water samples in to freeze them, lots of filters and that sort of thing.
Perfect, thank you. And another Rebecca asks, where in the water column do students collect samples?
Yeah, so for safety concerns, all of the students do shoreline surface water sampling. And it kind of varies based on what infrastructure is available at these different lakes. Some might be off a dock, some might just be on the shoreline itself.
And are there particular depths that the samples are collected?
They're just all surface water.
Oh, okay, gotcha.
Yeah.
Thank you. What age students are you working with?
These have all been high school students, and it ranges from sophomores all the way through seniors. So any age of high school.
Perfect, thank you. We're whizzing right through these. Okay. Have you identified any benthic cyanobacteria in the lakes or reservoirs that you are studying?
So because we're just collecting surface samples and we're not doing algal counts, we have not. We are just looking at chlorophyll A instead of doing any kind of speciation of the samples. We do collect phytoplankton samples just to have in case we see something really interesting that then later we could go back and do algal counts if we needed to.
Great, thanks. And it sounds like you said that students are and schools are also asking their own research questions. So there's some amount of flexibility there, it sounds like, in what students are looking at.
Yeah,
okay.
The students at Cheney were really gung-ho to just further this project, and they're doing a few other things there too, which is really cool.
Jennifer asks, how did you conduct your toxin assays?
We do ELISA kits for both of these toxins. And I think I included the detection limits on those slides as well.
Perfect. And for those of you that may not have been watching the chat box closely, those slides, as well as the presentation, including audio files, will be available on our website after the presentation. Lauren from the Upper Mississippi River Basin Association asks, are the results being published in your master's thesis? And she just expressed interest in the graphs that you displayed.
Yes, that is actually what I'm working on right now, and for the remainder of the year is writing up all of this research. And we hope to publish that as soon as I'm done.
Excellent, thank you. Jim asks, did you use a column sampler, a 2 to 6 foot column sampler at all for chlorophyll A?
The students are just collecting surface samples. So in a few of the locations, they're using PVC column samplers, but in some locations, they're just doing a grab sample because that's what they can do basically, given the infrastructure that's there.
Okay, so that's variable, and it sounds like you can handle that variation in sampling method.
Yeah, it's still just a surface sample, no matter what.
Okay, great. And Kelly, I know Kelly Mottman and Rebecca North were communicating about getting a HABs program, a ROS program started where Kelly is, so that's great. And again, that offer stands for other folks that are have high schools that are near lakes and might be interested in this project. High schools in any state in the US, so that's very cool. Lynn asks for Emily, was there any attempt to differentiate chlorophyll A results by cyanobacteria and true algae? And she's wondering if the chlorophyll A totals seem relatively stable, but the contributors changed, if you looked at that at all.
So, like I said, since we didn't do algal speciation, we have those phytoplankton samples where we could go back and look at that at some time. We were just looking at chlorophyll A here. And I see there's a follow-up question on if we attempted to correlate phycocyanin concentrations. And so I wanted to add that actually this last year, we added phycocyanin to this project. And so we have filters that the students filtered out that we will run phycocyanin tests for them, but we just haven't gotten there quite yet. So it will be interesting once we're able to see how much phycocyanin is there relative to these parameters too.
Thank you. And yes, Joe is the answer. You can get the copy of the presentation when it's uploaded shortly after the presentation, within a week, and probably sooner than that. Okay, Amy Shields offers up, and I think this was during Melissa's presentation, that EPA Region 7 has videos from their HABs prize and challenge last year. If those could be resources, I think that we could spread the word about those resources being available. So I'm guessing Melissa's taking notes on that right now.
Yeah, absolutely.
And Don asks, what were the nitrogen and phosphorus concentrations in these lake waters? Could they be tied to sources, farm facilities, failing septic tanks, etc.? And have you or in the lab done any watershed-based research to show what surrounds the lakes being studied?
Yeah, so both of these systems are pretty high nutrient systems, and question. The Cheney watershed has a lot of agriculture in the region, and Bethel Lake is in more of an urban area. And there are definitely projects ongoing, looking at how land use affects water quality too. But we did not look at that specifically in this project.
Never a loss for work to do, right? Okay, Dawn asks Melissa, is there a list, and questions keep popping in so that I lose my question here. Melissa, is there a list already of resources to which we can refer and therefore not duplicate? So what resources do we already have, and where is that list?
Sure. So no, we don't have a list already developed. It's just something that we saw when we were looking for information. This goes back to the methodology of the original phase 1. We had a lot of questions, you know, whose resources necessarily are we compiling for this analysis? And we just noticed while we were doing a lot of the research that we saw that EPA, that HABARCA, and other agencies had a lot of great information that was available. So you know, when it comes to the major agencies, we'll be able to find EPA's information on cyanos. We'll be able to find, you know, HABARCA and others like that. But if you have resources that you want to make sure we include, just feel free to send those to me. We'll sort through duplicates, and it's really no problem. In terms of, you know, what we have lists of, we have the references and citations included in the white paper, and then we have again, that inventory that we conducted was all extension produced products.
Yep, so there's definitely room for other products, and I see Dawn has put an EPA resource page there to make sure that we get the resources. So thank you. Yeah, we, as I think Melissa indicated, we did really focus for the purpose of this project from a time perspective, but we definitely want to, you know, we know that there are a lot more resources out there, as Melissa said, and if we can help amplify and increase access to those resources, we certainly want to do that. Kathleen, I think Kathleen, we've answered your question about what that surface water meant. I think I had that question too, and I hope that Emily clarified what that surface water sampling actually means. David, and if she, please feel free to follow up on your question. David asks, how does the sample collection protocol change in the winter when there is ice cover?
Yeah, so yeah, just to clarify, the students are doing grab samples from the top water, basically. And in the winter, especially for those northern lakes where they might have pretty thick ice cover, we do provide them with an ice auger so they can auger through the ice. This is kind of on a case-by-case basis where we work with the teacher to make sure that they are going out with the students to sample just to keep everything as safe as possible, and then the students sample from immediately below the ice.
Perfect, thank you, Emily, and thanks, Amy, for those contact names there for us to follow up on additional resources that we might want to access. Okay, let's see. Oh, Greg is saying his students have broken through the ice to collect samples. So yep, multiple ways to get that done. And Dawn posts another resource, a PDF of do-it-yourself tests to help determine if algae is BG or not. What is so? Thank you. I was like,
yeah, no, that's awesome, Dawn. I really appreciate that. This is a conversation that we had when we were talking about developing the fact sheets about, you know, it is really questionable when people are looking at algae to what kind of algae is that? What color is it? You know, is that subjective? So having that resource is helpful because it's definitely the type of thing that we talked about when we were looking at this. So I'm looking at that webpage right now, and I appreciate you sending that in.
I got the DIY, but I could not figure out the BG on the fly. So thank you for that. Okay, any other questions for our presenters before we do a quick wrap up here? We have another request for you. So you betcha. So don't go away before I get a chance to give you this request. So he's seeing no other questions. I'll keep watching there while we finish up. Okay, so we wanted to let you know and ask you to participate with us. The North Central Region Water Network is launching a What for Water Twitter campaign to increase awareness around what we all are doing for water around the region. So this is just a fun thing. What for water? What are you doing for water? And it's a way for us to feature the important work that's going on related to water across the region. So if you have something you want to share with us, and you all have been great at doing that on this in the chat box here, the discussion has been great. And we do, sometimes in webinars you sit there and absorb information, and we love the fact that you're being interactive with us in here and providing your expertise to everyone on with us today. So thank you so much for doing that. We want to continue that through this What for Water Twitter campaign. So to participate, you can send the following to Grace Hirschberg, our amazing communication student, at the email there. And Grace, you can put your email in the chat box. That would be awesome. It might be easier for people to copy. And we're looking for responses by July 20th. And what we would like is a one-sentence description of a program, resource, or action you do professionally for water, or in your personal life, that's fine. And one sentence about why you think it matters. And a photo would be great if you're particularly the photo of you doing the thing with water. But if you have a different photo, just of you, that's fine too. And your Twitter handle, if you have one, or your organization's Twitter handle, if they have one, we'll do that to feature you and your organization and give you that credit. So that's our invitation. We would love to hear from you by July 20th. And again, Grace put her email in the chat box. That's Hirschberg, Berg with an ERG at wisconsin.edu. So thank you so much for your time today and for the great conversation. Please follow up with Emily, Rebecca, and Melissa with Emily and Rebecca North, if you're interested in having a site near you become a ROS site. And Melissa, if you have any feedback on or contributions to some of those outreach strategies and tactics that we shared with you today. Thank you so much. Thanks to our speakers for excellent presentations. And again, please share the recording of this webinar when it's up on our site with folks that might be interested that weren't able to attend today. Have a great afternoon, everyone, and you take care. Thank you.